All right. Hi, guys. So this is should be your second genetics lecture. If you'll take a look at my screen, so you should have already opened this notes packet. You should have already listened to lecture number 1. If I open up the notes packet, it should look like this. You should have already listened to the first lecture and filled in all this. And last thing we did together was completed the practice set number 1. So that should be completed in your pink packet. And then we're going to move on today with not that much new stuff. We're actually just going to look at the ratios here and then Mendel's laws of inheritance. So that's where we are. Please make sure you're using your notes. I don't know how we can still be confused by where the notes are. Anyways, okay, so we are there in our notes. We're going to keep rolling with kind of analyzing Punnett squares. So far what we've looked at is with the results of the Punnett square, we have gotten good at writing the genotypes. So that, for example, if we look at the offspring here, the possible genotypes are big B. Here, that's the genotype showing the alleles. These individuals are big B, little b, and then this individual's genotype is little b. The phenotypes are what they physically look like. So 75% of them are going to be purple. Purple is the phenotype, and then 25% of them would be white. To take it a step further, you may be asked to show the ratio, so the phenotype or the genotype ratio. If we start with the phenotype ratio, so the phenotype ratio is just the probability of all the possible phenotypes. There's only ever 2 possibilities, so it's always expressed with 2 numbers. Okay, so if you look here, we show the number of the dominant phenotype first, colon, because it's a ratio, and then the number of the recessive phenotype. In this instance, remember, phenotype is the color. So there were 3 purple individuals, which is the dominant phenotype. So we start with a 3 and a colon 1, because 1 individual down here would be the recessive phenotype. Let me emphasize how important zeros are. I think I talked about it in the last lecture, that zeros here are just as important as any other number. So you'll see some problems where there will be like 0 that have the dominant or the recessive, and you put that there. So you put a 0 in the ratio. The genotype ratio is looking at the possible genotypes, but there's 3 options. So the genotype ratio will always have 3 numbers, even if one of those numbers is 0, right? Because 0 is just as important. So we always start with the dominant first, just like we saw with the phenotype ratio, the dominant was first. So we're going to start here with the homozygous dominant. So the number of individuals that have the homozygous dominant genotype. And then the number of individuals that have the heterozygous genotype, and then the number of the homozygous recessive. So in this same cross, we have 1 individual that has the homozygous dominant. So we start with a 1, colon 2, because there are 2 individuals here with the heterozygous, colon 1. So there's 1 individual that has the homozygous recessive. Okay, so let's do a practice here. Okay, a homozygous black guinea pig falls in love with a homozygous brown guinea pig, and all their babies are big B, little b. So, here I have filled in the parents. So the homozygous homozygous black, let's do that first, is on top. So big B. And then the homozygous brown is little b, little b. If we fill in the square, we see the potential offspring. All of them are going to be big B, little b. And that's because this parent can only offer a big B, and this parent can only offer a little b. So everyone, even if they have 300 babies, all of them will be big B, little b. So now I'm asking for the ratios. So the genotype ratio and the phenotype ratio. So the phenotype ratio, there are all four are going to have the dominant phenotype, right? So in this case, they're all going to be black. So 4-1, meaning there's 0 chances of a homozygous. Sorry, I was just reading homozygous. The recessive phenotype. If I let that go away, the genotype ratio. So you can see here that the number zeros are so important. Okay, so with the genotype ratio, 0 of their babies are going to have big B, big B. So we have to put a 0, colon 4, because all of their offspring are going to be heterozygous, and then 0, because there's 0 chances of having a homozygous recessive offspring. Let's do another example. A heterozygous brown-eyed man fell in love with his science partner in biology class. 10 years later, he walked down the aisle with this lovely blue-eyed lady. What color eyes will their future children have? So if we make the square, he is heterozygous brown. So he's heterozygous, he's big B, little b. And she is blue eyes, which is recessive. So she must be little b, little b. If we fill in the square, we see big B, little b's. So half big B, little b's and half little b, little b. If we fill then the genotype and the phenotype ratio, the genotype ratio, remember there's 3 possibilities with genotype. So there are 0 offspring that will be big B, big B. 2 of them would be expected to be big B, little b, the heterozygous, and 2 of them would be homozygous recessive. Remember, phenotype only has 2 numbers because it's either the dominant or the recessive phenotype. In this instance, these individuals will express the dominant phenotype, which is brown eyes, and these individuals will be the recessive or the blue eyes. So 2-2. Okay. Moving on in your notes, we're going to just kind of quickly look at Mendel's laws. Mendel really produced, his work really supported 3 laws, and one of them is the law of dominance. And this really just states that dominant alleles can hide recessive alleles. And we've seen that already with even just our simple little Punnett squares. So the dominant allele hides the recessive. The law of segregation states that the gametes, or sex cells, remember from last unit, are formed with only half of the chromosomes. We see that with meiosis, right? The number of chromosomes in our sex cells gets split in half. That's what we call the law of segregation. The law of independent assortment states that the gene pairs are segregated into those gametes or into those sperm and egg randomly and independent of each other. So when we talked last unit about meiosis and reproduction, the collection, due to crossing over, the collection of chromosomes in every single sperm and egg is totally random and independent of each other. There's no rhyme or reason. And that's what Gregor Mendel stated with the law of independent assortment. Okay, and I think the last thing is a test cross. So something, and this was on the air test, so weird, but by golly, they threw one at him, I think it was last year. So a test cross is used to determine if an individual with a dominant phenotype is homozygous dominant or heterozygous, right? So looking at me with my brown eyes, you couldn't tell if I'm homozygous dominant, so I might be big B, or I might be big B, little b. In order to kind of figure out, we can do what's called a test cross. So on inspection of the heterozygous, a heterozygous is indistinguishable from a homozygous dominant. So what we do is always cross the unknown with a homozygous recessive, so little. So if you look here, let's say that this is my brown eyes. Let's look at the cross here on the top. Let's imagine maybe I'm big, right? So maybe I'm big A, and I have two dominant alleles for my eye color. We're going to cross the homozygous dominant with the recessive allele, and it's always with the recessive, and here's why. If I had 100 babies, if you look here, no matter how many babies I'd have, they'd all have brown eyes, okay? Because they'd all be big A, little a. If, however, scroll down here, if I'm actually heterozygous, and instead of big A, I'm big A, little, I get crossed with a homozygous recessive, and we cross it with a little only because that's the only genotype we're actually certain of. If someone, for example, in this case has blue eyes, we know they're recessive for a fact. So there's no way that anything funny could happen in a test cross. Sorry, so down at the very bottom, let's imagine now that we think I'm heterozygous, so big A, little a. I get crossed with a little a, have lots of babies, and we would expect half of my babies to be brown eyes and half of mine to be blue, right? So if I'm heterozygous, we would expect that trait, the recessive trait, to pop up again when I have babies, even though it's hidden in me. So with a test cross, we always take the homozygous dominant and heterozygous and cross them each with a recessive, homozygous recessive genotype. Here's a question, or here's a practice. So in one species of kangaroo, tall ears is dominant to short ears. There's a kangaroo with tall ears hanging out in your backyard. How would you determine if he was heterozygous or homozygous? So the only possible way is to actually cross them. Now, this seems crazy when it's like humans or frankly big old kangaroos, but this is what Gregor Mendel did with his pea plants, right? So he picked pea plants because they had lots of babies, they were easy to cross, and so by God, he actually bred, he intentionally crossed individuals and looked at their offspring. So let's imagine the kangaroo here. So this kangaroo has tall ears, big T. We don't know by looking at him in my backyard if he has big T, or big T, little t. So the only option here is to cross him with the one known genotype, which is short ears. So little t. So here I set up my crosses. The little t's are on the side. So we're going to physically find a short-eared kangaroo and cross them and let them have some babies. And then we're going to look at their offspring. One possibility is that this tall-eared fella is big T, but again, he could also be big T, little t, and that's what we're hoping to find out. So let's say they have babies, and we see their offspring. If all their babies have tall ears, there's a really good chance that he then must be homozygous, right? If you look down here at the bottom cross, sorry, that silly thing keeps popping up. If he was heterozygous, that means that he passed on the little t recessive allele to half his babies. So we would expect then the offspring to, about half of them to have tall ears and about half of them to have short ears. Okay, now pause the video, pull out your pink genetics practice set packet, and follow along with me as we go through some practice problems. If you're at home and you haven't gotten your papers yet, just jot this down on a piece of scrap paper, and just so you have it for your reference. Okay, so practice set number 2 is actually question 4. So a common anaconda is typically olive green with large brown spots or small brown spots. You can see large brown are dominant. If a heterozygous male mates with a small spotted female, what will the genotype and phenotype ratio be for their offspring? So I'm going to make my cross and go back to the question. And I said a heterozygous male. So he's going to have big, little. And again, I mentioned last unit to make your letters look like really obviously different. So I have a big L and a little l. He's heterozygous. Mates with a small spotted female. And so she must have 2 recessive alleles. So 2 little l's. If I fill in my little squares. Whoops. And I put phenotype first. So my phenotype ratio, remember phenotype only has 2 options. They either have the dominant phenotype or the recessive. In this case, 2 individuals would it be expected to have the dominant phenotype. So I'm going to put 2. And 2 would be expected to have the recessive. So the ratio would be 2 to 2. In my genotype ratio, remember there's 3 options. So first we look at how many individuals are homozygous dominant, and there's none that are big L, big L. So remember, put a 0. You've got to put that 0. The next number is how many individuals are heterozygous, big, little. So I have 2 here that are heterozygous. And then the last one is homozygous recessive. And I have 2 individuals here that are little l, little l. So my phenotype ratio and my genotype ratio. Let me just show you what so many of you want to do. You want to forget about the 0 in the, in any of the ratios really. But look what happens. Let's imagine I didn't put a 0. For the genotype ratio, that looks totally wrong, right? First of all, we don't know what it's 2 of, but also I'm going to immediately count it totally wrong because to me it looks like a phenotype ratio. Okay, so make sure you're putting your zeros. Okay, if you need to pause and rewind, do that as many times as you need. And I'm going to move on to the next one. Okay, anacondas have enormous jaws attached by super stretchy ligaments. Big S, that allow the animals to swallow prey whole, or some have tight ligaments, little s, and may try for smaller prey or have to eat in the water to help them swallow. Females are much longer and stronger than males and typically get to choose their mate based on a courtship ritual underwater. If a hybrid female chooses a heterozygous male with stretchy ligaments, predict the genotype and phenotype ratios of their offspring. Okay, so I'm going to set up my little cross. And a hybrid female, remember hybrid is the same as heterozygous. So she's going to be big S. And I make my weird little cursive S's so they look totally different. So she's hybrid, so heterozygous. And he's heterozygous as well. So he's going to be big S and little s. So let's go ahead and fill in our squares. And the phenotype ratio. Remember, there's only 2 options, the dominant. These 3 individuals are going to have the dominant phenotype because they have 1 dominant allele. So 3. And 1 individual here will be recessive. So 3 to 1 is the phenotype ratio. And, genotype ratio. Remember, we start with homozygous dominant. So how many individuals are big, big? Here we have 1. How many are heterozygous? We have 2 individuals. And homozygous recessive is 1. Okay. All right, last one. Anacondas can have eyes on the top of their head or a little lower on the sides, which is recessive. which determines how well they can see when swimming at the surface of the water. A herpetologist has a beautiful female with eyes on the top of her head, but she wants to know if she's homozygous or heterozygous. So she decides to perform a test cross. Show your results. Okay, so I'm going to try to fit my two crosses here. So remember, she has the dominant phenotype. So we look at her and she's eyes on the top of her head. So we know she's has at least one big T, but we don't know if she's big T, big. We don't know if she's big T, or big T, little t. So to do a test cross, we cross this individual with a homozygous recessive, because we know by looking at that anaconda, because the eyes are on the side, we know her genotype, their genotype right off the bat. So we're going to take this female that's either homozygous dominant or heterozygous and breed that anaconda with an individual that has eyes little on the sides of their face. Okay, so let's fill in the blanks. No. Okay. And when they have babies, what we would expect to see is if the individual is homozygous dominant, then we would expect all their babies to have the dominant phenotype, right? So all dominant. If they have babies, and you know what? About half of them have the dominant and half of them have the recessive, we know that that individual must be heterozygous. So this is what we call a test cross. And again, test crosses seem silly when it's humans, but again, even a snake would have, lots of babies in just one clutch. So it is something that even conservation scientists that are trying to, successfully manage populations would do test cross. Oh my gosh, isn't that hilarious? That is funny, you guys. Okay, all right, that is it. Now you should go on to some of your practice.